All right, let's talk about sampling with replacement and making a new population with old allele frequencies. So here we have, for the at-home lab, a couple of beans. We've got the small red beans, and we've got 50 of those that I've poured out and counted. And we've got the Great Northern beans. They had a better PR, I guess, and there's 50 of them. So we're going to say that this is a single allele. Maybe it's a single egg. And we're going to put that into the gene pool, which is this paper bag. I've cut the top off the paper bag to make it easier to reach my hand in there. So we're going to make 50% of these white beans here, these Great Northern beans, and that's going to be an allele frequency of 0.5. So we're going to say P is 0.5 because the frequency of that dominant allele represented by the Great Northern beans is going to be 0.5. And the frequency of the recessive allele, as represented by these red beans, is going to also be 0.5. It is a 50-50 mix of beans in the bag. And I'm going to shake that up. And now I'm going to randomly reach in there. And I'm going to take two alleles to make a diploid individual. So here we go, two alleles out. And what do you know? That's two of the lowercase. So I would write down that under my genotypes of the homozygous recessive, I'd write down 1. And I'm going to put these back in the bag. Why do I put them back in the bag? Well, I took these guys out, and now there are 48 red beans and 50 Great Northern beans in there. So if I've changed the allele frequency by removal. So in order to sample from the same population accurately, I got to put them back in. I'm going to shake it up a bit, and then reach in to another random sample. This time I got another one of those. So I'm going to record that. And I'm going to repeat this as many times as my instructor tells. That is sampling with replacement. Now, let's say I have a allele frequency here, and I've got an allele frequency that doesn't match my 50-50. So what I'm going to do is I'm going to pour these out. And I've got a 60-40, so 60 white and 40 red. So what I'm going to need to do is I'm going to need to take 10 red out of here. That's 5 red. 1, 2, 3, 4, 5, and another 5 red. So I'm removing 10 red. So now there are 40 red and 50 white. So now I'm going to add 10 white here. 1, 2, 3, 4, 5, 6, 7, 8, and 9, 10. Beans. There we go. So now I've recreated this population. This population now has 60 white beans and 40 red beans to better represent the sample I got. So that's going to be the next generation. I'll remove these red beans, put them in a pot, eat them as soup, and I'm going to pour these back in, and my new population now has a different allele frequency. So I've changed that allele frequency. Maybe I want to turn it to 70-30, in which case I'd take 10 more red beans out of there and put more white beans in. Shake it up, and now I'm going to start sampling with replacement again to see what the new population is like. Oh, look, a heterozygote. And you would do that as many times as your instructor requires. So there you have it. Sampling with replacement and changing the allele frequency of a population to match an allele frequency of a sample. Thank you.